Good morning. This is Michael Dong speaking on episode number 19 on UHPLC 1, where we describe overview, perspective, and benefits. We welcome LCGC International as a channel collaborator. Here is our agenda. First is perspective, where we describe glossary, historical perspective, and the modern HPLC platform with higher performance. Second is UHPLC concepts. Besides high pressure, there's a need for low dispersion and low dwell volume because of the low void volume and peak volume of the UHPLC column. Also talk about instrumental bandwidth and the UV flow cell design. Finally, the benefits. There are three major benefits: very fast separation with good resolution, very high resolution analysis for very complex samples, and how to facilitate rapid method development using UHPLC, and also other benefits. This video came from data from several white paper from the chapter of the books, chapter 5 of the book, and 3 of the white papers published in 2017. Here is some terminology and acronyms for UHPLC. Number 1, UHPLC stands for ultra high pressure LC, typically describing system with pressure limits above 15,000 psi. Also, the system come with low dispersion of low instrumental bandwidth and also lower dwell volume. Now, UHPLC should not be confused with UPLC, which is a very common misconception, because UPLC, standing for Ultra Performance Liquid Chromatography, is a trademark from Waters, meaning UPLC, meaning Waters Acuity System, which debuted, or is the first commercial system debuted in 2004. So if you say UPLC, you really means Waters Acuity System. Others are column void volume, which are defined here. The liquid whole volume of a column, roughly about 50 to 65% of an empty column volume. Peak volume is the contingent on the column volume. Also, you can measure it by multiplying the peak width with the flow rate. Later on, we defined system dispersion, which or instrumental bandwidth relate to the fluidic volume of the system flow path as seen by the sample only. Other detect the flow cell volume, extra column band broadening caused by system dispersion, which are deleterious or harmful to the column efficiency, particularly for smaller ID columns. Dwell volume is defined as a system volume from the point of solvent mixing to the head of the column. It is much responsible for the gradient delay volume. Dwell volume are typically smaller, definitely smaller, for high pressure mixing system for binary pumps. Here is a slide describing historical perspective on UHPLC. Before 2004, HPLC resolution has been bounded by system pressure of about 6,000 psi, thereby limiting the practical performance of column efficiency to about 20,000 plate, or a peak capacity of about 200. Now, everything changed after 1997 with the proof of concept research published by Professor James Jorgenson. Here's a picture of Professor James Jorgenson in 1997. So currently, UHPLC is a modern HPLC platform capable of much higher system pressure, anywhere from 15,000 psi to 22,000 psi. Also, in conjunction with lower system dispersion to reduce extra column band broadening, and also lower dwell volume to reduce gradient delay time. And typically at the debut is used with sub 2 micron particle with an ID in the diameter of 2.1 mm ID when it was introduced. In 2014, David Guillaume, who's a professor at University of Geneva, and myself, we co-edited a special issue on UHPLC. Okay, the issue is labeled as UHPLC, where we are 10 years after the commercial introduction. It was an ACS journal called Track, one of the top reviewed journal, and containing 19 invited papers on the scope and also the where we are 10 years after the commercial introduction of UHPLC. Here is the publication in Liquid Chemistry in 1997 and 1999, published by Professor James W. Jorgenson and also his graduate students. Right away, one can see an outrageously impressive performance, right? A plate count of 190,000 plate in about 10 minutes, which is definitely record-breaking. And the peak capacity, which is also equally impressive under grading conditions, a peak capacity of 300 in about 30 minutes, right? So this is the first proof of concept research by Professor James Jorgenson. Now, the first commercial system actually debuted 7 years after that 1997 paper. And I was told that Professor Jorgenson actually was a consultant with Waters. And Waters in 2004 have a breakthrough introduction of the Waters Acuity UPLC, which is a trademark, registered trademark for ultra performance LC, 15,000 psi. A few years later, some of the major manufacturer, the four major, also introduced their version of UHPLC, shown here, right? So a little bit on the later models. The UPLC H model, which is the quad pump, the first one's a binary pump, was introduced in 2010. Actually, this is actually the top seller for Waters. They also make the I class, the M class for micro LC, ARC system, which is a intermediate pressure system, dual path system. Also the Acuity Plus and the Acuity Premier using the max peak technology. Thermo Fisher Scientific introduced the Vanquish in 2014, which is still the highest rated system in UHPLC. They also have the Flex system, 15,000 psi, and the Vanquish Core and Dual. Agilent came quite a bit later, about 6 years later, introduced the 1290 Infinity 1 system, 18,000 psi, and follow on with Infinity 2 and Infinity 3 in 2014 and 23. And they also have the 1260 and 1260 Prime, which is a quad system, a lower pressure system. And Shimadzu Nexera also introduced a UHPLC called the Nexera series in 2010, follow on by different second generation and third generation systems. Some of the more detail can be found in this white paper published in 2017. First paper of the series is UHPLC 1, Perspective, Instrumental Features in LCGC magazine. This table is a list of pressure rating and the maximum flow rate of the four major manufacturers for UHPLC. Most of them also make a biocompatible version or bioinert version made from either alloy, titanium, or PEEK. This figure came from chromatogram courtesy of Waters Corporation, explaining why higher pressure are needed, primarily by column packed with smaller particle column. So here there's a comparison of 4 different columns of the same length, which is 15 millimeter or 5 centimeters. First one is packed with 10 micron. The next one is 5 micron, 3 micron, and 1.7 micron, a sub 2 micron. Now, obviously, when you use a column packed with smaller particle size, the efficiency are much greater because the minimum plate height, h, is roughly equal to 2 times the particle size. So n is equal to the length of the column divided by twice of the minimum plate height. So for a sub 2 micron column, 5 centimeters long, you get 15,000 plates, while 10 micron column give you only about 1/6 of the column efficiency, resulting in less resolution between 2 and 3. You can see it's much better. And also a higher sensitivity to peak height because of the higher column efficiency. Now, unfortunately, part of the physics is the fact that the pressure drop of the smaller particle column is proportional to 1 over dp squared. So by taking down the column diameter 6 times, the pressure drop of the same length of the column is increased by 36 times. So that's the reason why you need a system called ultra high pressure LC with higher system pressure. We also mentioned something called lower system dispersion of instrumental bandwidth to reduce extra column band broadening, because typically using a smaller column with the smaller inner diameters and a smaller peak volume. So to prevent extra column band broadening, typically by the flow cell, we need a system with less system dispersion. To measure system dispersion, in this case study shown on the Agilent 1290 Infinity, one would disconnect the column from the system and rejoin the two ends with a zero dead volume union. So when you inject a small volume of caffeine solution here, right, under these conditions, under very high data acquisition rate, one would get a trace of the instrumental bandwidth shown here. Here for this 1290 system, the 4 sigma bandwidth is equal to roughly about 16 microliter, which is quite small in comparison with HPLC system. A typical HPLC system is much larger. And here is a tabulation of the various systems from acuity, the Agilent 1290 with switching valve, the thermal vanquish, and also HPLC systems such as Agilent 1200 with a bandwidth of 30 microliter, and Waters Alliance with a bandwidth of 45 microliters. Here's a table of system dispersion system measure. Either came from my own data or data that are published data. So once again, Waters Alliance system 45 microliter, and various. The lowest one is actually documented from the Acuity I class with 4 to 6 microliter of 4 sigma instrumental bandwidth. Okay, one of the things that allow the instrumental vendor to make a system with low bandwidth is the introduction of a very small flow cell. Now, one can make the flow cell small, but it's difficult to make a flow cell small with pretty long path length, because one required to have at least 1 centimeter long path length so that sensitivity under UV absorbance detector would not be reduced. And the way Waters achieved that in 2004 is to introduce a patented flow cell called the light-guided UPLC, where the flow cell is actually made from a Teflon tube, specifically a Teflon AF tube because of low reflective index. So you can have a total reflecting flow cell, meaning that all the lights in this very, very tiny flow cell is completely 100% totally reflecting in there because of the low refractive index. The Teflon AF actually have a refractive index that's lower than water, not the regular Teflon. So this flow cell have a path length of 10 millimeter and also a volume of 500 nanoliter or half a microliter, which is significantly smaller than a regular HPLC type of UV detector, have a flow cell of about 8 microliter to 10 microliter. Now, with the smaller flow cell, there's an opportunity to make a high sensitivity flow cell using a longer path length, right? So waters also support a flow cell with 25 millimeter path length. This figure illustrates how do you measure dwell volume. This case study is the dwell volume measurement by using this procedure, which is being documented and published in chapter 4 of the book, using this operating condition, right, where we fill mobile vase A with water and B water spiked with 0.6% acetone. This mobile vase actually give you a absorbance unit of about 1. And then you run a very fast gradient in about 5 minutes, going from 0%. And once again, you disconnect the column and reunited the two ends with a 0 dead volume union. And when we get a trace of the profile of the gradient, and to measure dwell volume, basically you extrapolate the linear gradient with the baseline. And in this particular UHPLC, there's a choice of 2 volume of a mixer, right? This is a binary pump, and it does require a mixer. The mixer used is called a jet weaver, which is a high-tech mixer. The standard size is 35 microliter, and when you flip the jet weaver mixer, you have a larger mixer of 100 microliter mixer. Typically, for high sensitivity application, we would use the higher volume 100 microliter mixer here. So if you use the standard mixer and determine the dwell volume, one get a measurement of 140 microliter, which is quite small. Considering the dwell volume of the HPLC, such as the Alliance of 1200, would be about 1 mil, right? So it's significantly smaller. And that allow you the opportunity to do high throughput screening because of the lower gradient delayed time and delay volume. This slide actually summarizes the UHPLC benefits. 3 major benefits. Number 1 is very fast separation with good resolution. Second is higher resolution separation for very complex sample. The third one is rapid method development of HPLC method using UHPLC system and columns. Other benefit are flexibility and also high sensitivity if one uses the extended path length, and also higher precision performance methods. The first benefit, faster analysis with good resolution. Here we illustrate a publication by Professor David Guillaume in 2008 on European Journal of Biopharmaceutical Analysis, where he compared a conventional HPLC column gradient condition separating 12 pharmaceutical compound, right? So one used a C18 column, 15 centimeter long, very typical of a HPLC system and column, packed with 5 micron material. The optimum flow rate is 1 mil per minute of this column. The injection volume is 20 microliter, and runtime is about 45 minutes, right? So this is very typical gradient separation of 12 drugs. Now, in comparison, Dr. Guillaume actually used something called a geometrically scaling. When you scale it to a smaller column, a shorter column with smaller ID packed with smaller particles, you do something called geometrical scaling. And the flow rate is also have to be adjusted, and the flow rate should be 0.2 mil per minute. However, because of using smaller particle size, the optimum flow rate is actually 0.61. So he optimized it using the optimum flow rate with geometrical scaling of the other parameter. And when you inject, because of a smaller column volume, you have to inject less, right? So the injection volume is 1.4. However, you can see that when you compare the two performance of the chromatogram, one have very comparable performance, almost exactly the same type of resolution, the same type of sensitivity pKa. However, we are injecting much less sample because the column is less. And one get a runtime of about 5.1 minute, and the final peak actually eluded about under 3 minutes, right? So the outcome is 9 times faster analysis with similar resolution using UHPLC. So this is obviously much more preferred, right? Much faster, same resolution, same sensitivity. Another case study published by myself in a paper called A Universal Generic Reverse Phase Gradient Method for Pharmaceutical Analysis in 2016. I documented a very quick generic method in about 2 minutes, which is very much compatible to most of the pharmaceutical assay and can be adjusted very quickly to give you a stability indicating assay. And shown here is the separation of 12 NCE or new chemical entities in about under 2 minutes. And I'm using both a QDA, a single quant mass spec, with a UPLC from Waters, which allow me using Empower to actually pull out the mass spec and also the UV spectrum. So this is a very nice generic method that most people can adopt very quickly. A second benefit is high resolution separation. This case study came from Brian Yang of Bristol Myers Squibb. He actually gave that presentation at New Orleans HPLC 2014, showing a drug product with 2 APIs, which have their, this is the retention marker mixture of the expected impurity in degradation product, right? So using in the QD charge surface hybrid, phenyl column, 150 millimeter long, 2.1 millimeter ID packed with 1.7 micron. So a very high resolution column, very simple mobile phase, acyl nitrile, and mobile phase A is a TFA in water, and one can achieve a very impressive resolution. Next one is even more impressive example of a very complex pharmaceutical. This one is a stribial oral tablet formulation with 4 API. This one, this application came from Gilead, from Daniel Prudhomme of Gilead Science, in a joint publication in 2014 in LCGC magazine, and myself. The stribial tablet have 4 API, and the system suitability mixture consists of up to 60 peaks. Very impressive separation using a very simple gradient, a 5-segment gradient program to do that. This method was actually developed with the help of Fusion QBD, which is a software program using DOE, a designer experiment to optimize the condition of this gradient program. This case study demonstrate rapid method development, right? Here is a column screening study of a force degraded sample, right? The problem is some of the sample actually collude with the API, very easily colluted. And a column screening quickly determined that the best column to use is a polar embedded column rather than a C18 column or a Fanu column. Fanu column actually did the worst. The highlight is the fact that UHPLC column screening can expedite method development because we're using a very short column. And under 30 minutes, we can do a complete screening using 3 or 4 columns. This example was published in 2014 by myself and also Kelly Zhang of Genentech, called UHPLC Method Development in Track magazine. The other benefit is flexibility and resolution, right? One can almost customize it by using different column length coupled with a different gradient time. So here one can use a fast method. This is a separation of that forcibly degraded sample. When you optimize the gradient, you can use the fast method using a 5 centimeter column and a 6-minute gradient. One can get separation of these 4 major peaks from the forcibly degraded sample. And one can see that at the end of the gradient, we have 3 peaks over here. Now, in contrast, you can also use a high resolution method, which is using a much longer column, coupled with the longer gradient time, right? Typically, with long column, you need a longer gradient time. And you can see that the 4 major peaks are much better separated. What is actually very impressive is the fact that if you look at the last 3 peaks, one can actually see that it's really not 3 peaks, it's actually 5 peaks, because there's probably a pair of diastereomer being separated by the high resolution in coupled with higher gradient time. So under these conditions, and the condition is actually, the time is actually not that long, right? So the gradient is only a 20-minute gradient, right? So this is capable of so you have flexibility, you use very fast and also very high resolution condition without sacrificing too much longer analysis time. And this slide demonstrates higher UV sensitivity using an extended path length flow cell. So with the smaller flow cell, one have the option of using a extended path length flow cell. Shown here is the max light flow cell from Agilent, which is a 6 centimeter long flow cell, meaning that if you want to have high sensitivity, you can do so by injecting larger amounts and also using this max light flow cell. So under the same condition, one gets 6 times higher signal noise ratio with the max light flow cell. Now, unfortunately, this is not very much used typically because when using area normalization, this might become a problem because the API peak can very easily saturate it. So for applications such as a cleaning verification where you use external standardization rather than internal normalization method, this is actually much more desirable for those applications, but unfortunately, not for stability indicating method, because using these type of flow cell, you can the main peak gets saturated very, very easily. Here's my list of references. The key one are the 3 white paper published in 2017, either in conjunction with Davi and also or as a sole author, and also the chapter 5 of the book, and also the special track issue of the 18 paper on UHPLC, where we are 10 years after commercial introduction, and also a few other papers. On that note, we want to summarize and conclude. We talk about perspective of UHPLC, glossary, historical perspective. We say that it's HPLC with a higher performance platform. We mentioned several concepts, meaning that the UHPLC is a low dispersion system with high system pressure limit. Concept which are important is the void volume of the column and the peak volume, the resulting peak volume of the smaller ID column packed with smaller particle. And also the concept of instrumental bandwidth responsible for extra column band broadening, and the design of UV flow cell give you small volume, low dispersion, but with adequate path length, and also lowered gradient dwell volume. The benefits are very fast separation with good resolution, very high resolution for complex sample, particularly a drug product with multiple APIs, and the facilitation of rapid method development. On that note, we ended our episode number 19, UHPLC overview, perspective, and benefit. We thank you for your attendance. And once again, we welcome LCGC as a channel collaborator and urge you to subscribe to this channel for future videos. See you next time.